Hello everyone, welcome back to my EasyGrade YouTube channel. Today we are going to start the second part of the lesson Microorganisms, Friend and Foe. Now, first we will study about cleaning the environment. Bujo and Pelli, the two school students, observed their gardens making manure and they collected various types of plant waste, vegetables and fruits from nearby homes and gardens. Okay, this waste was then placed into a pit for disposal. After some time, it was transformed into a manure. They wanted to understand how this transformation occurred. Means the process of waste turning into manure is explained by the action of microorganisms. These tiny living beings break down complex organic materials and releasing essential nutrients back into the soil. Okay. In contrast, they also observed that some materials like pythogen bags, empty glass bottles, and broken toys placed in a separate pot, means pot B, did not undergo any such change. This shows that microorganisms are unable to decompose these types of materials. Okay, and microorganisms play a crucial role in the environment. They decompose large amounts of dead organic matter, such as dead plants and animals found on the ground. Okay, this decomposition converts the complex dead matter into a simpler substances. These simpler substances are then utilized by other plants and animals, completing a natural cycle. Okay, this process of effectively helps to degrade harmful and fuel smelling substances, thereby contributing to the cleanliness of the environment. Now see the activity 2.5, demonstrating decomposition. They took two pots and soil, and plant waste, means leaves, fruit peels, vegetables, scraps, etc. And the last one they took uh, polythene bags or empty glass bottles, broken plastic toys. And I will tell the procedure how to do this activity. Take two pots and fill each pot halfway with soil and clearly label the pots as A and B. In pot A, place plant waste. In pot B, place items like pythogen bags, empty glass bottles, and broken plastic toys and set both pots aside. Observe the contents of both pots after 3 to 4 weeks. After 3 to 4 weeks, you will notice that distinct difference that plant waste is pot A will have significantly decomposed, likely appearing dark, crumbly, and reduced in volume, resembling manure. Okay, the items in pot B will remain largely unchanged, and the organic plant waste in pot A is biodegradable, means it can be broken down by living organisms, and the items in pot B are non biodegradable, means they cannot be broken down naturally by microorganisms. Okay, next you will find that plant waste is in pot A has been decomposed. How did they happen? The plant waste in pot A decomposed due to the activity of various microorganisms like bacteria and fungi. Naturally, it is present in the soil. You can see bacteria and fungi. These microbes feed on the organic matter, breaking into down into a simpler inorganic substances through a process called decomposition. Okay, microbes feed on the organic matter, breaking it down into a simpler inorganic substances through a process called decomposition. And the contents of pot B means the pathogen, glass, plastics will not decompose. This is because microorganisms do not have enzymes or mechanisms to break down the complex chemical structures of these synthetic materials. Now, see the harmful microorganisms. While many microorganisms are beneficial and play various roles in the environment and in our lives also, it's important to recognize that some are harmful. These harmful microorganisms are responsible for causing a variety of diseases in humans, plants, or animals. And these microorganisms are called pathogens. This is a key term. Pathogen is a simple and microorganisms that cause diseases. These are the bad guys. We need to be aware of that. These are responsible for spoiling our food, damaging our clothes, and unfortunately causing illnesses in humans, animals, or plants also. So, knowing about them is crucial for protecting ourselves and our environment. Okay, now see the disease causing microorganisms in human, that is communicable diseases. Now, how do these tiny pathogens get into our bodies and spread? They can enter through the air we breathe, the water we drink, and the food we eat. And this also disease can spread through direct contact with the infected person or even through carriers. When a disease can be transmitted from an infected person to a healthy person through air, water, food, or physical contact, we call it as communicable diseases. These are this is a very important definition. So highlight it in your textbook. Okay. For example, common cold. We all had it, right? Common cold. When someone with a cold sneezes or coughs, tiny droplets containing the virus spray into the air. If you are nearby, you might inhale these droplets and catch the cold. This is why covering your mouth and nose when you cough or sneeze is also important. It's a simple act that prevents the spread of infection. Okay, look at the little cartoon on your page that illustrates this. Cholera, chickenpox, tuberculosis, these are the other example of communicable diseases that spread through different pathways. Now see the carriers of diseases causing microbes. Now let's look at the right side of the page where it talks about carriers. These are organisms, usually insects or animals, that transmit pathogens from an infected sources to a healthy one without necessarily getting a sick themselves. They are like disease messengers. For example, you can see the housefly. Imagine a housefly, it sits on a garbage, animal waste, or contaminated surfaces, picking up thousands of microbes on its legs and body. Then it flies and lands on your uncovered food. It just transferred through those diseases, causing microbes directly to your plate. This is why it's so to keep our food covered at all times. And the mosquitoes, these are even more dangerous carriers. The female Anopheles mosquito, as shown in a figure 2.8 on your page, carries the parasite that causes malaria. When it bites an infected person, it pick up the parasite and then transmits to it to the healthy person through its next bite. And the Aedes mosquito is known to carry the dengue virus. So, mosquitoes are not just annoying, they are serious disease vectors. Okay, let's move on to the next page. Here we have a very important resources, table 2.1, some common diseases caused by microorganisms. This table is like a quick reference guide showing you the microbe responsible, how it spreads, and how to prevent it. Now, common human diseases and prevention. Quickly go through some of this, highlighting key points. First one, TB, caused by bacteria spread through air. Prevention involves keeping the patients isolated and vaccination. And measles and chickenpox, both caused by viruses spread through air contact, isolated and vaccination are key. And next, polio, caused by virus spread through air or water. Vaccination is crucial. India has made great progress in eradicating polio due to widespread vaccination campaigns. And next, cholera and typhoid, both caused by bacteria. Cholera spreads via water, food, typhoid by water. Prevention focuses on good personal hygiene, sanitation, eating properly cooked food, and drinking boiled water. Vaccination is also available for typhoid. Okay, and hepatitis A, caused by virus spread through water. Drink boiled water and get vaccinated. And malaria, caused by protozoa spread by mosquitoes. Prevention includes using mosquito nets, spraying insecticides, and preventing water collection, which is just discussed. Okay. So, looking at this table, what's the biggest takeaway for preventing many of these diseases? Cleanliness, vaccination, or safe food, water, controlling carriers. These are our best lines of defense. Okay, and the next one is diseases caused by microorganisms in animals. It's not just humans who suffer from microbial diseases, animals too. Okay, anthrax, this is a very dangerous bacterial diseases that affects both humans and cattle. And foot and mouth diseases, these are viral diseases that severely affects cattle. These diseases can devastate livestock and impact agriculture. Okay, and the next one is diseases caused by microorganisms in plants. And guess what? Even plants get sick. Microorganisms cause diseases in many important crops like wheat, rice, potato, sugarcane, orange, and apples, etc. These plant diseases can significantly reduce crop yields, when means less food for us and economic losses for farmers. We will see specific examples on the text page. Now look at the table 2.2. Common plant diseases means microorganism and mode of transmission. Just like we looked at human diseases, understanding plant diseases helps us protect our food supply. One is citrus canker. This disease affects citrus fruits. It's caused by bacteria and spreads through air. You will see lesions on the leaves or stems and fruits. Next one is rust of wheat. This is a measure fungal diseases for wheat crops. It's caused by a fungus and spreads through the air and also through seeds. If infected seeds are planted, the disease can quickly spread throughout a field. Okay. Now the third one is yellow vine mosaic of bindi, okra. This is a viral disease affecting okra plants. It's caused by virus and spread by insects. The leaves develop a yellowish mosaic pattern, reducing the plant's ability to photosynthesize and thus reducing the yield of okra. So, these diseases highlight how microorganisms can be a significant foe to our agriculture, impacting food production. Okay, now we will study about the food poisoning. Bojo getting sick after a party. Why do you think that happened? For bad food, uh, spoiled food, germs. You are absolutely right, no? Food poisoning is caused by microorganisms growing on our food. When these microbes grow, they produce toxic substances. Even small amounts of these toxins can cause severe illness, including vomiting and severe causes, etc. Okay, it can even be fatal. These emphasizes why food prevention is so critical. We need to stop these harmful microorganisms from growing on our food in the first place. Okay, now we will study about the methods of food preservation. Okay, the first one is chemical method, and that salts and edible oils. These are traditional preservatives especially used in pickles. They create an environment where microbes struggle to grow. Okay, and the second one is sodium benzoate and sodium metabisulfate. These are common chemical preservatives used in commercial products like germs and squashes. They effectively inhibit microbial growth also. Okay, and second one we will study about preservation by common salt. Salt is an ancient preservation method. It's used for meat, fish, raw mangoes, and so on. How does it work? Salt draws out the moisture from the food. Microorganisms need moisture to survive. Okay, so by dehydrating the food, salts inhibits their growth. Okay, and next we will study about preservation by sugar. Think of jams, jellies, and squashes. Sugar is used here as a preservative. Similar to salt, a high concentration of sugar reduces the moisture content, making it difficult for bacteria to grow. Okay? And next, preservation by oil and vinegar. This is a common for pickles, oil creators, and barrier. Cutting off air supply and vinegar makes the environment highly acidic. Okay, most spoilage causing bacteria cannot survive in such conditions. And the next one is heat and cold treatments. These are the methods we use daily, like cold treatment. First one is cold treatment. Keeping food in a refrigerator slows down microbial growth significantly. Cold temperatures inhibits their metabolic activities, thus preserving food for longer. And next, heat treatment, pasteurization. Okay, look at the bubble on your page. Why does the milk that comes in packets not spoil? And what is pasteurization? Pasteurization is a process where milk is heated to about 70 degrees Celsius for 15 to 30 seconds and then suddenly chilled. These rapid heating and cooling kills most of the harmful microbes present in the milk without affecting its nutritional value or taste. Okay, this revolutionary method was discovered by Louis Pasteur. Okay, and next we will study about storage and packing. Nowadays, many dry fruits and vegetables are sold in sealed airtight packets. This method prevents the food from coming into contact with air and moisture, which are essential for microbial growth. This keeps the food fresh for longer. So, these are the various techniques we use to protect our food from the microorganisms. Which method do you think means you often uses it at home, and which method do you use? Comment me in the comment box. Okay, and next we will study about nitrogen fixation. You might remember learning about the bacterium rhizobium in earlier classes. These amazing bacteria live in the root nodules of leguminous plants like peas and beans. Okay, as shown in figure 2.9 on the text page, what do they do there? They take nitrogen gas directly from the atmosphere and convert it into a usable form for the plant. This process is called nitrogen fixation. Okay, I will repeat: nitrogen gas directly from the atmosphere and convert it into a usable form for a plants. This process is called nitrogen fixation. Okay, and it's vital for plant growth because nitrogen is a key component for proteins and other essential components plants need. We will explore the entire nitrogen cycle in a more detail on the next page. Now we will study about nitrogen cycle. Our atmosphere is made up of about 78% of nitrogen gas. That's a huge amount of nitrogen. But here's the catch: plants and animals cannot directly observe and use this atmospheric nitrogen. Okay, it's in a form that's not accessible to them. This is where microbial friends come to and to save the day. Let's trace the journey of nitrogen using the diagram figure 2.9, the nitrogen cycle. Okay, first one is atmospheric nitrogen. This is a starting point, the vast reservoir of nitrogen gas in the air. Okay, and second one is nitrogen fixation. This is a process where atmospheric nitrogen is converted into usable nitrogen components. Okay, in that nitrogen fixing bacteria like rhizobium in root nodules of leguminous plants. Okay, and blue green algae present in a soil perform this conversion. Okay, and lightning also contributes. The intense journey of lightning can convert atmospheric nitrogen into nitrogen oxides. Okay, which then dissolve in rainwater and fall to the earth, enriching the soil. Third one is compounds of nitrogen in the soil. Once fixed nitrogen exists as various components like nitrates in the soil. Okay, next uptake by plants. Plants absorb these nitrogen compounds from the soil through their root system. Okay, this is how nitrogen enters the food chain. And fifth one is animals eat plants. When animals consume plants, the nitrogen compounds are transferred to the animals' bodies. Okay. And the sixth one is nitrogen's waste and death, means decomposition. When plants and animals die, or when animals excrete waste decomposers, various bacteria and fungi in the soil break down this organic matter. They convert the complex nitrogen compounds back into a simpler ones in the soil, releasing ammonia. And and the seventh one is nitrification. Other bacteria then convert this ammonia into nitrates, which plants can again absorb. Okay. And the last one is denitrification. Finally, special bacteria called denitrifying bacteria converts some of these nitrogen compounds back into the gaseous nitrogen, which then returns to the atmosphere, completing the cycle. So, the nitrogen cycle is a continuous, beautiful process where nitrogen constantly moves between the atmosphere, soil, and living organisms. All thanks to the incredible work of microorganisms. Without this cycle, life as we know it wouldn't be possible, because plants wouldn't get the essential nitrogen they need for growth. Okay. And this is about this lesson. Thank you for watching this video, and if you have any doubt, please comment in the comment box. I will clear your all doubts. And if you like this video, don't forget to like, share and subscribe to my EasyGrade YouTube channel. Thank you.